Next we have from the Hauptman-Woodward Institute, Dr. Sarah Bowman.
Okay, thanks very much. Thanks very much for organizing this. I think it's been great. The morning session was great, and this afternoon's been great so far as well. So I'm going to tell you all a little bit about some of the things that we're doing at HWI in the High-Throughput Crystallization Screening Center, and a lot of the work that we're excited about starting. So first of all, just as an introduction for anybody who is not familiar with us, we are in Buffalo, New York, and we are a center that is really available for anybody to use. So you can send your samples from anywhere in the world for us to set them up for very sparse screening. We actually set up in one tray instead of 96 conditions, 1,536 conditions. We use a microbatch under oil method, and this is just an example of one of the trays that we set up. And for microbatch under oil, what we do is we add oil to each well, and then 200 nanoliters of the chemical cocktail, 200 nanoliters of your protein, and then we spin those down to merge the drops under oil and have a robust imaging schedule over time to monitor the crystal growth. And so this is obviously a little cartoon of what that looks like. So one of the things that is a little bit different for us is that our crystals grow really only at the bottom of the plate in the merged drop that's under oil. And so we have to image our plates upside down. And so we turn the plates over, and our imaging technologies to actually detect crystals, we use both imaging tables for visible images that have been made here at HWI, and we also use a Rock Imager 1000 with SONICC. This is really our workhorse for detecting crystals and especially detecting nanocrystals, which we'll talk about as time goes on here. And Formulatrix was great in actually setting up adapter trays for us to be able to put our trays in upside down, as well as be able to image the 1536, because that's a little bit unique relative to a lot of the imaging that occurs in other labs. And for us, what we really want to be able to do is to provide these crystallization services for people who may not have access to some of these robotics and automated techniques, as well as imaging methods. And so again, at the end, I'll share our website information. So one of the things that is interesting about doing such large wells, such large plates, so many different conditions, 1,536 conditions that are imaged 7 times using the visible images, once using UV-TPEF and SHG, which we'll talk about in a second, is that for each sample that we set up, we actually generate a minimum of 13,824 images. And so that's a lot of different images to actually go through. And you'll hear a little bit about how we use the different methods here to really pull out the things that are generating crystals. So just really briefly, what does SONICC get you? SONICC in our Rock Imager 1000 has two additional features beyond visible imaging, and one is the UV ultraviolet 2-photon excited fluorescence, which allows us, just like any UV imaging, to detect concentrated protein. So that will detect protein crystals, as well as proteins that are in gelatinous states, precipitated, but it's dependent on the protein actually having tryptophans or aromatic residues. And we'll see some examples of this in just a second. SONICC also gives us second harmonic generation, which is something that will identify chiral crystals. And so these two photons, which are from the same laser source as the UV-TPEF, generate a second harmonic generation signal. Positive signals here are high contrast and white against a black background. They will detect most space groups. And so what's really great about this is that they can detect submicron crystals or nanocrystals, which are very helpful for things like X-ray free electron lasers. So again, what we have is, oops, I went a little bit too far. What we have is our 1536 conditions. We image these visible UV-TPEF and SHG, and how can we use these multimodal images to actually detect protein crystals. So first of all, it makes identification of protein crystals relatively straightforward. And what you're going to see in the next number of slides are a series of images where we have visible UV-TPEF and SHG images. In this case, it's actually quite clear that we have protein crystals in all three of our modalities. We can also use it in images of our wells that may not be as clear that these are protein crystals. Again, in this case, we can clearly identify that these are protein and that they are crystalline. Not all crystals will give a positive SHG signal, so there are some space groups that won't light up in SHG, but again, we can see visible and UV-TPEF. And even sometimes when the crystals may be below precipitate, we can see the bright spots. And in this case, we can actually rule conditions out. So this actually helps us a lot. Negative conditions are helpful. And we can also identify probable salt crystals. So in this case, we have these lovely little crystals that we wouldn't want to optimize because though they are definitely crystalline, they have no UV-TPEF signal. So we can also detect nanocrystals. So this is, again, really exciting for anybody who is doing work with X-ray free electron lasers where we need very small, homogeneously sized crystals and a lot of them. So in this case, you can just barely see the crystals here, but using the UV-TPEF and SHG, we can identify these crystals. This is one of my favorite examples. This is one of the only actual well in this 1536 tray that generated any crystal hits. You would not necessarily identify this as crystalline, but using the UV-TPEF and SHG, we can see that these are actually beautiful nanocrystals that are growing very nicely. So I want to just end the last few minutes with some new advances. So one of the things that I know that Mayank already talked about was that there is a new photomultiplier detector for enhancing the SHG signal. This is, again, really helpful for detecting nanocrystals. This is an example of one of the wells from one of our 1536 trays that we actually sent out to Formulatrix where they tested for us how well it does. And really, we do see an enhancement of about 5 times for most of the wells that we might not otherwise identify crystals in. And I'm going to end by talking about some new work that we're doing in collaboration with scientists at SSRL and LCLS to actually do some room temperature sample mounting and actually do in situ crystallization to be able to develop crystallization that we can then ship to a synchrotron or to an XFEL source and able to set it up directly onto the beamline without any manipulation or handling. And so the goal here was to actually generate a robotic compatible crystallization plate. And it has actually already been implemented. You're going to see some prototypes in just a second. Essentially what we do is these wells, there's a number of different wells that we can set up, and each of these wells can contain a variety of different types of pins. This is one of the pins that they have developed at SLAC, and essentially you can set up, as you can see, hopefully a large number of different crystallization conditions. You can then image these wells and be able to know exactly where your crystals have grown, mount them directly onto the beamline and be able to collect data on these. And there's a variety of different kind of compatibilities. What's really cool about this is that we can actually ship these things across the country. So first of all, there was an initial setup for how the different how these might be shipped, and these were developed by some interns at SLAC, at SSRL, to be able to figure out how we could actually set this up to be able to ship room temperature crystals with really no manipulation. So this was the prototype, and actually we tested this about 2 weeks ago. This is now the prototype. So here's an actual image of the tray in the blue box here, and they're being set up in a dewar, mostly to keep it upright. So it really requires a Hampton-style magnetic base. If the sample base fits in this little tester that's shown in Figure B, then it will fit within the plate. And so in this situation, we can actually set up crystallization, a variety of different conditions. We can store these plates. We can have them humidity controlled. We essentially put the mother liquor in below this with some agar, so it doesn't, it's not actually liquid, but it keeps it humid. And in fact, we've shipped samples from California to here at HWI in upstate New York, and then ship them back. And actually the crystals last beautifully. They do wonderfully, and I think I'm out of time. So I know some of the other developments, I believe Janet's going to be talking about the Marco project. We're also doing some other things, and with that, I will say thank you for your attention. Our website is www.getacrystal.org, so you can look us up if you have further questions. Send me an email. Thank you.